This last video is a wrap-up of our phylogeny section. Um, we've gone into a lot of detail. We've talked about a lot of different aspects of phylogeny and phylogenetic inference. In this one, it's going to be shorter, and we'll mainly just look at why phylogenies are so important. And the readings, especially paper number 6, I think it was 6. Anyway, the Hillis paper. No, it was paper 5. Anyway, the Hillis paper talks about why phylogeny are so important, why they are a foundation for all of modern biology. And so the short answer is they give us a roadmap of species relationships. And because species are connected by evolution, that's a critical aspect for all parts of biology. So here's an example of one phylogeny that we've looked at easier, two competing hypotheses, right? Are elephants or all placental mammals, the elephant just as a representative of that, are they more closely related to the marsupials, or are the marsupials more closely related to the monotremes? And this says something critical about the evolution of live birth, right? So if this is the correct hypothesis, which by the way, almost all the data now supports this is very well supported as being the correct hypothesis. That says this transition from laying external eggs to an internal embryo happened once. We can map it on there one time. And that's a very different story about the history of these organisms than if this was the correct set of relationships. Then either eggs in Platypus and Echidna, the monotremes, would have had to evolve separately from the eggs in these ancestors, or egg laying would have had to have been lost, right? A transition to internal fertilization would have had to happen 2 separate times. So these are critical for helping us understand the history and the characteristics of these organisms, which is the purpose of biology. And so here's a more in-depth phylogeny of the same thing. And notice we have very strong at a level of 100, very strong support for the placental mammals and the marsupials together in a single group. Now, once we have good, strong support, we name these. We give these classifications and names. So the name of that group is Theria. Eutheria is the placental mammals. Theria is the placental mammals plus these marsupials. Okay? So these are great. We can test them. We do them. And as I mentioned earlier, is that for many, many hypotheses, many phylogenies, we have very well supported hypotheses. Many of them now are getting into the level of theory where we know them very well. We can also include extinct species onto our phylogenies. Now, the only issue with extinct species is that the data is limited. Usually we have bones, maybe shells or other hard material. Occasionally we have a little bit more information, but usually not much. And so although we can include extinct species, data is somewhat limited. However, so here's an example of a reconstruction of fossil horse skulls with, we have good enough information. We can look at teeth and the bones and the organization of all these different types of characters in the skull. So that's really great. But can we use DNA? And the answer is, for most fossils, no. DNA has an upward limit, although it's a very stable molecule. It has an upward limit maybe in the hundreds of thousands. Some people argue 1 million or a few more, but really there's not any DNA left after millions of years. So we can use DNA for some fossil species. So if we dig up a mammoth that's been locked in the permafrost, really good conditions for DNA storage, then we can very likely pull DNA out of it. In fact, there's an ongoing project to try and de-extinct or de-extinctify, I don't know, anyway, the mammals. Look it up, it's kind of cool. It's, kind of like Jurassic Park, but Jurassic Park, even though it kind of started the whole idea of bringing animals back to life, is impossible because there's no DNA that goes back more than 65 million years. But woolly mammoths, woolly rhinos, maybe, right? Because they've only been extinct for tens of thousands of years, and there's still DNA that is recoverable from well-preserved specimens. Now, there's still many, many hurdles, and it's unlikely that we'll see it in the near future, and maybe we'll never see it. But DNA can be used for other things than de-extinctifying, and we use it very readily for phylogenies. So while people are trying to bring back woolly mammoths, others have used their DNA and put it into analyses to figure out how they are related and fit them into the tree of life. As we find new fossils, we revise, change. This is the great thing about science, right? You get new data, you can add it to your analyses, and that might reinforce what you've already established, or it might revise it, it might change it. So this is a recent fossil that is very useful in helping us to understand the transition from lobed fin fishes to the tetrapod vertebrates, the amphibians, then eventually mammals, and so on down the line. And this was discovered in Greenland and added to our body of knowledge. Now, we've already talked about phylogeography. Let's just look at a couple of examples. We know from geology and also now from biological studies that about 250 million years ago, there was one supercontinent. You already know this. This is a very common, well, it's usually well communicated, and you learn it in grade school, and very well accepted now. 250 million years ago or so, continents started to break up. It was a long, slow process. We ended up breaking up the northern continents, which is called Laurasia here with shallow sea in between. And then the southern continents were all locked into one thing. By the way, notice India. This is kind of cool. I don't know if you knew this. India was part of Gondwana. It was part of the southern continents. But the Indian plate migrated and eventually plowed into the Asian plate. And the reason that we have the Himalayas, the highest mountains on Earth, is because India is still smashing into the Asian plate and pushing up those mountains. But anyway, southern continents in one mass, northern continents in another one. And many species that have a low ability to disperse, that can't get around very much, still show remnants of this ancient geographic separation between the northern continents and the southern continents. So it's present in stoneflies. The species you find in stoneflies in the southern hemisphere are very, very different from the northern hemisphere. But you don't know stoneflies. I don't know, maybe you do, but probably not. You look at it and like, I don't know stoneflies. But this is one maybe you're more familiar with. So the Ratites, which are these large bipedal running on the ground, and they're flightless birds, right? All birds really are bipedal, but these are the flightless ones. Ostriches, rheas, emus, cassowaries, and then the kiwi in New Zealand. They all occur only in the southern hemisphere, right? At least until people started bringing them to new places. But that's where they're endemic to. That's a good vocabulary word. Endemic means native to an area and found only in that area. So rheas are endemic to South America. Ostriches are endemic to Africa, emus in Australia, and so on down the line. And so this distribution, because these guys couldn't fly and couldn't distribute and migrate, this distribution is a remnant of the ancient, the ancient arrangement of the continents. So sometimes geography is locked into the relationships of organisms. These are all from a common ancestors. The Moas, by the way, are an extinct group from New Zealand. There are many, many species of Moas. They were much larger even than our ostriches, but were hunted to extinction by the early Maoris. Imagine having a 12-foot turkey walk through your camp. That'd be pretty hard to resist. Anyway, but showing you remnants of that ancient history. But other things too, not just distribution. We can say, does a nocturnal character, does that evolve once or twice? Are there characters that are associated with one another? So notice in this analysis, the evolution of arboreal habitat, living up in trees, is correlated with the evolution of being nocturnal. So living up in trees and being nocturnal. And that's a cool little thing. And then scientists may even begin to ask, well, which came first? Did these organisms move up into the trees and then become nocturnal? Or did they become nocturnal and then move up into the trees? And with this data set, we can't answer that. But if we were able to find additional species that maybe had one character and not the other, and put it into the phylogeny, that might help us to understand the evolution of this thing. So we can do virulence of viruses, we can do distribution of organisms, we can do, are you nocturnal or not nocturnal? We can look at, does color evolve more quickly than other characteristics? And over and over and over again, we use phylogenies as the beginning point for all of these analyses. Here's one other. Organisms often have many of genes in the same gene family, and those arose from a single ancestral gene. So early vertebrates had gene duplication events, and so they ended up with 4 opsons. Opsons are genes that code for a protein in your eye, and that protein allows you to see wavelengths of light. So if you have multiple opsons, then there's the potential for those opsons to see different wavelengths of light. Now, humans have three opsons that allow us to see color. We have a fourth that's just for sensing light in general, but we have three color opsons: a short wave, a medium wave, and a long wave. And together these all work to give us our range of different colors we can see. Some organisms have different opsons that allow them to see into the ultraviolet, and some even up into the infrared, although that gets less common, at least for eyes. There are other things like heat pits and snakes that allow infrared sensing. But many see much farther down into the ultraviolet than we can see. But for us, this is what we see. But we're actually much better visually than most mammals. Almost all other mammals have, including most primates, but there's some that are closely related to humans that have three. But most mammals have two. Birds, lizards, all the reptiles have four. And so when we look at this, it's tricky, but as we add more and more species, we've realized that the vertebrate ancestor of all vertebrates, it's a little redundant, the ancestor of all vertebrates had four opsons and probably had pretty good visual spectrum. But they were lost. Maybe they weren't important in the mammal lineage. This may even be the synapsid lineage, although we don't have opson data for extinct species. So we lost two of our opsons. Mammals and our ancestors included only had two opsons, but somewhere in a portion of the primate lineage, us included, we regained an opson. So we have a brand new opson. So this medium wavelength is an opson that resulted from a gene duplication that is not present in these other lineages. So that's a really cool story, and we only know it and know the details of it because of the phylogeny. And so we can find convergences, right? If two organisms that are distantly related share a common feature, it's very obviously convergent. We can say, no, it's not convergent, it's homologous, right? This is a trait that they gained from a common ancestors and did not gain independently. And so if you are given a phylogeny and a character, you should be able to map that character onto the phylogeny and then tell me whether it is convergence, simplesiomorphy, or simple homology, right? So look at that. If those terms are still unfamiliar, review. Meet me in one of the Zoom discussion sessions and we can go over it again, but you need to get to that level where you can recognize that given a tree and characters. And some characters are very, very obviously convergent. So there's a place in New Mexico called the White Sand Dunes. It's actually now a national park. And there are many, many species, including lizards and mice, but other species that have evolved very light white coats, white skins that allow them to blend in. And that's obviously, you wouldn't say this lizard is now close related to this mouse because it's lizard. It just has this one characteristic. So obvious convergence. But sometimes convergence is not as obvious, and we really need a phylogeny to help us figure it out. So swimming in pelagic zones, these deep water oceans where organisms pretty much swim all the time, many of these organisms have energy-saving mechanisms where they only move the hind part of the tail. Sharks, pelagic sharks, and these tuna fish do it rather than moving the whole body back and forth. And that is a convergence. And we know that because when we map it onto a tree, it happens separately. Okay, so review homology. Make sure you don't confuse it with convergence, which is a type of an analogy, right? And make sure you understand simplesiomorphy. So here we have convergence, a trait that happens twice, right? If it happens once and is lost in some of the descendants, then it is a simplesiomorphy, okay? Now, for the purposes of time, am going to skip Perology and Orthology. It's a little bit more of an advanced topic, and we don't need to look at it. But basically, it's looking at gene families and understanding relationships among genes and gene families. If you're disappointed, you can come and see me. We can talk about it. It won't be on exams, though. Or you can just sign up for my upper division biology course where we'll talk about it in much, much more detail. Okay? So a couple final examples of how phylogeny has been helpful. I picked these. There are hundreds upon hundreds that I could choose, but I picked these because they're kind of fun and interesting, and then we'll finish with a couple of key concepts here. So for a long time, it was thought that venom in snakes evolved separately from venom in lizards. There are a couple of venomous lizards. Now, just to be clear, venom is a toxic molecule that's made to be injected or put onto the skin or onto the surface of an organism. It's basically either a defensive or an offensive weapon that gets injected into the target. Poison is a toxin that's kept in the body, and it's there as a defense or a deterrent to being eaten, right? So if you eat something and you get sick and die, it's a poisonous object. If something bites you and you get sick or die, then it is venomous. So that's just the distinction. And it was thought that venom in snakes, which is a very highly derived, advanced, quite complex, it was thought that evolved separately from venomous lizards. There's one small group of lizards that was known to be venomous, the Gila monster, and their close relatives. However, once people made a phylogeny, they realized that snakes are actually more closely related to some lizards than they are to others. So snakes are really just highly derived, advanced lizards, much like birds are highly derived, advanced dinosaurs. And some of the closest relatives of the snakes included this venomous lineage, the Gila monsters. So they reevaluated and they started looking at some of these others, the varanids in particular, but iguanas also, and they realized that these guys had some, although not very toxic, had some of the same toxins that were used in venoms in both of these, and that the Gila monster and the snake had many of the same genes that were components of their venom. And so that led to this idea that, well, maybe venom did not evolve separately in these groups. Maybe it evolved ancestrally and then was passed down. And so they did a reevaluation of that, and sure enough, it looks like that's what happened, is that venom evolved early in the history of this lineage of lizards. Some of them have very, very mild or even have lost venom, but the snakes developed it in addition to losing their legs and becoming snakes. The snakes also developed these very highly complex venom systems and injection methods. And we wouldn't know that story, and we wouldn't have been able to investigate it without the phylogeny to give us guidelines. Now, finally, let's talk about why we lose traits. This is a very common thing and is maybe even inevitable. When a characteristic is very, very useful, the organisms that carry it will do very, very well. They'll diversify and will usually end up with hundreds upon hundreds, maybe even thousands or hundreds of thousands of species that carry those characteristics. And inevitably, as time goes by, some of those species are going to be exposed to environments where the very feature that made them so successful is no longer an advantage. Eyes, for instance. Eyes are incredibly useful as they've allowed for diversification of animals in general, but many of the animal groups. However, when an animal, a species, gets locked away in a cave or in an area where the eyes are no longer useful, inevitably, eventually, those eyes will go away. It's just because random mutations happen all the time that remove eyes from individuals. If you're a species that needs those eyes, then those traits never get passed on or very rarely get passed on to descendants. However, if you're living in a cave, that's maybe even an advantage, right? You lose your eyes, it doesn't matter because it's dark. But not only that, that energy that was being used to build and develop eyes can now be diverted and used for other things that are maybe more important in a cave environment. And so we see traits lost all the time. Snakes lost legs. Many other groups have lost their legs. This cave salamander from central Texas has lost its eyes. Other groups, fish, insects living in deep caves without any exposure to light also lose their eyes. And so it's very common to see these traits that have been lost. And we call these traits vestigial traits. Good vocabulary word. You need to know it. Just a couple examples in humans. Sometimes we see multiple examples. The coccyx is a good example of a vestigial trait in humans. We no longer use it. It's really not, doesn't serve any purpose, but it's a remnant of a once important ancestral trait, our tail. So we'd call that our tailbone. My favorite example of a vestigial characteristic, one you should be familiar with in humans, are the erector pili muscles. Now that's a technical term. The more common term is goosebumps. These are little tiny muscles at the base of the follicle that cause your hair to stand up on end. Now, what is that for? Why do we get goosebumps? Well, think about what are the situations when we have goosebumps? And we get them when we're cold or when we're under stress, like scared, right? Or in a fight and all our nerves go on edge, our hair stands on end, right? So what's the purpose? Well, it turns out that in humans, there really is no purpose now. They are vestigial, they're a remnant. But in our ancestors, they served a purpose. So think about it in other animals. A cat's a good example. Polar bears are a good example, right? So when your cat is scared, when it's challenged, its hair stands on end, and it makes it look bigger and a little bit more scary. That's dynamic, that's useful and helpful for them. Polar bears, when they get cold, their hair stands on end. They trap a little bit more air against their body, and it helps them to stay warmer. So those are the two purposes for erector pili muscles, which are no longer relevant to humans, right? I don't know any humans that are hairy enough that when their hair stands on end, they look bigger and scarier, or that would keep them warmer, right? So another good vestigial trait. But all organisms at one level or another have vestigial traits.